Hi, hello, good morning everyone. I'm Dr. Harika on behalf of Vista's Learning. I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the pre-recorded videos, live sessions, and also you're going to get an access for all the study materials, assessments, concept videos, animated videos. Moreover, you're going to get this lecture notes in the form of PDFs. Get your acquaintance with all the channels available and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, make sure to click on the like and share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Saying that, let's begin our today's session. We are discussing the chapter Molecular Basis of Inheritance, which comes under the unit Genetics and Evolution. Right? So, in the previous session, we had a discussion about the transcription unit. Different types of RNAs like rRNA, tRNA, mRNA. And also we discussed about the process of transcription that is happening in prokaryotes as well as in the eukaryotes. In the present session, let's discuss about the genetic code. So, here how the genetic information that has been carried by the mRNA, where it is actually present, genetic information is stored in DNA. From DNA to mRNA, that is RNA. From now, RNA is going to give an information how the sequencing of the amino acids has to be there in the structure of proteins. So here, before understanding the translation, that is how the sequence of the amino acids has to be placed to form the structure of the proteins, we need to understand genetic code. We have been discussing about replication, transcription, translation. During replication as well as replication and transcription, right? So, replication is all about making a copy, replica of the DNA by using DNA as its template. So, here the template is DNA and the produced strand is also DNA. So, here the enzyme, that is DNA dependent DNA polymerases, is useful to synthesize newly formed DNA. And that process we call it as a replication. So, replication where double-stranded structure is going to uncoil and form a single strands. Now the single strands of the DNA will act as a template for newly forming strands. That means template as well as newly formed strands. New strand, both are of DNA type. That means nucleic acid and the produced end product is also again nucleic acid. That is, information is carried from one nucleic acid to another nucleic acid. Here both are DNA. Now coming to the transcription, where from this DNA it is going to be transcribed into mRNA. So here DNA, from that copying of the genetic material, genetic information is happening to another nucleic acid, that is mRNA. And this process is called as transcription, right? This is translation and this is transcription. The point here is during replication and as well as transcription, here this is replication and this is transcription. So from here the mRNA is now further translates into protein. So before that, during replication as well as transcription, A nucleic acid was copied to form another nucleic acid. In replication, that is DNA to DNA, right? And in transcription, from DNA to RNA. Here the point is, both are nucleic acids. DNA as well as RNA are nucleic acids. This happens during replication as well as transcription, right? Now, hence this process are easy to conceptualize because the copying of the genetic information is happening from the nucleic acid to another nucleic acid, or from the nucleic acid to the nucleic acid. That is in the case of replication, DNA to DNA. Whereas in the case of the transcription, that is from one nucleic acid to the another nucleic acid, that is from DNA to RNA. But here it is all about nucleic acids. That means the biochemical nature is same, and that is nucleic acid. Hence the process are easy to conceptualize on the basis of complementarity. There is an existing complementary base pairing that present in the DNA as well as RNA. So it's very easy if we consider that this is a sequence which is present on the one strand of DNA. Then on the other strand of the DNA, T, C, G, C, G, C. We understood and we are able to write the sequence of the other newly synthesizing DNA strand because complementarity exists. And if you find to form the newly strand mRNA, that is RNA, then only exception that whenever the thymine is present in the DNA, here it is replaced with a uracil. But there exists a relationship in the form of complementarity. Here, the process of translation is a protein synthesis, requires transfer of genetic information. Even in replication as well as in the transcription, in both cases, genetic information is being copied down. In replication, genetic information is copied from one DNA to another DNA. There also complementarity helps us to form the new strand, that is new DNA strand. And in transcription, the genetic information is copied down from DNA to RNA, that is mRNA. Here in this case also complementarity will help us to synthesize a new RNA, right? But that is not the case that comes with the translation. After replication, transcription, here is a transcription. Protein. Here is a transcription. And here is a translation. We are talking about translation here. While replication as well as transcription is happening, the genetic information is transferring from one nucleic acid to another. And therefore, the complementarity will govern the process of transcription as well as replication. But if you come to the case of translation, where the information, genetic information has been copied from nucleotides, polymers of the nucleotides, to the polymers of amino acids, that is protein. So, here are the two biochemical natures. One is nucleic acid and other one is protein. So here, the process of translation requires transfer of genetic information from a polymer of nucleotides. Polymer of nucleotides to synthesize the polymer of amino acids. To synthesize the polymer of amino acids, that is protein. So here, neither does any complementarity exist between nucleotides and amino acids. nucleotides and amino acids. See, nucleotides, amino acids, polymer of nucleotides and polymers of amino acids. These are nucleic acids, whereas these are proteins. Protein and could exist between nucleotide and the amino acids, nor could any be drawn theoretically. See, in the case of nucleic acids, in the process of replication as well as transcription, if the purines are present like A, then T will come in the DNA. If G is present, then C, that is cytosine, will come in the DNA. That is how the complementary base pairing helps us to construct the new strand, either it is DNA or RNA. But that doesn't the same case with the translation, that is the synthesis of the proteins. There existed ample evidences though to support the notion that change in nucleic acids, if there is any change in the nucleic acids, if there is a change in the nucleotide, then it is going to show its effect by change in the amino acid. See here, change in nucleic acids, that is a genetic material, were responsible for change in amino acid, that is the structure of protein. If there is any change in the nucleus, a particular nucleotide, then that change will definitely show its influence on the change of the amino acid. If one amino acid is changed, it is going to change the structure of protein also. Now, this led to the proposition of a genetic code that could direct the sequence on what sequence amino acids has to come and combined in order to form a protein structure like this. Sequence of amino acids during the synthesis of protein. Out of the pool of the amino acids which are available in the cytoplasm, which amino acid has to come when? That information is carried by the genetic material, genetic information which is present on the mRNA. Now, if determining the biochemical nature, while we are discussing about the biochemical nature of the genetic material, whether it is a protein, whether it is a DNA, whether it is a RNA, it is very exciting, right? Biochemical nature of the genetic material and the structure of the DNA was very exciting. But here, the proposition and deciphering, arranging of genetic code were very challenging. Definitely, total number of amino acids is 20 different amino acids are present and we have only 4 nitrogen bases, 4 bases. How these 4 bases are going to code for 20 different amino acids that are coming in the structure of the protein? This is the question. So here, in a very true sense, it requires involvement of scientists from different branches, not only from a single branch. We require branches of different disciplines, from several disciplines like physics, biochemistry, and organic chemistry, like genetics. So scientists from different disciplines like physicists, organic chemists, biochemists, genetists, we worked together to decipher this genetic code. How? The 4 bases are going to code for total 20 amino acids that are going to appear in the structure of the protein. And it was Gamow. He is a physicist scientist, and here is a contribution by the Gamow, who argued that since there are only four bases, but it has to code totally 20 amino acids that has to appear in the structure of protein. Right? So, if they have to code for 20 amino acids, the code definitely should constitute the combination of bases. This is going to contribute a lot for the development of genetic code. So, out of 4 bases, some combination has to be worked out in such a way it is going to code for total 20 amino acids. So now, Gamow contributed in saying that the combination of the bases are required to code for total 20 amino acids. He suggested that in order to code for all the 20 amino acids, the code should be made of 3 nucleotides. It should be made of 3 nucleotides. See, we have 4. Definitely 4 bases can't code for 20 amino acids. If it is a we come to the conclusion that it has to be combined, combination of bases. If it is combined for 2 times, which goes like 4 into 4, again 16. We have still more 4 amino acids. So even this is not going to work out. Now, if it is 3 means 4 into 4 into 4, that is 64. This is more than enough to code for total 20 amino acids. This was a very bold proposition because of the permutation combinations, rearrangements, in which way it has to be rearranged such that 4 into 4 into 4 would generate 64 codons. Out of 64 codons, 3 codes for nothing. We consider them as a stop codons. And 1 codon will perform dual function, where it is going to act as a start codon, also called as initiator codon, and also going to code for a methionine, amino acid methionine. 64 codons generating many more codons than required. We actually require the codons which will code for 20 amino acids. But here 4 into 4 into 4, that is totally going for 64 codons, and this 64 codons are more than required to code total 20 amino acids. So now, here providing the proof that the codons were a triplet, made of, triplets means made of 3 nucleotides. Each codon is made of 3 nucleotides like this. 3 nucleotides, any possibility. And proving this is of daunting task, very difficult task. How to prove this? To prove that, there are so many scientists who performed so many series of the experiment. In that, the successful experiment was done by chemical methods, which was developed by the Hargobind Khorana, was instrumental. He was successful in synthesizing RNA molecule. What do you mean by synthesizing RNA molecule? See, actually, DNA will transcribe into mRNA, and this mRNA is going to translate into protein. But here the question is, how the nucleotides which are present on the mRNA are going to have the information, in which way the sequence of the amino acids has to come in the protein. To know that, he synthesized mRNA. He synthesized mRNA means he knows the sequence of the nucleotide. Once he knows the sequence of the nucleotide, then further whatever the sequence in which the amino acids are coming can be deciphered in such a way he could able to construct the genetic code. So synthesizing RNA molecule, that is mRNA molecule, which defined combination of, he know, because he constructed, he synthesized, he know the combinations of the bases which are coming in the mRNA, which are the nucleotides of homopolymers or copolymers. That combination he know. Now, by using the combinations of the polymers which are present on the mRNA, now there is a very well chances to decipher the sequence of the amino acids that are going to appear in the protein. Right? Along with him, Marshall Nirenberg's cell-free system. By using laboratory techniques, he is able to construct the cell-free system for protein synthesis and finally help the code to decipher. We now able to understand in what combinations that this nucleotides are going to sit, and that is coding for a single amino acids. In that, totally 64 codons, how they are going to code for a total 20 amino acids. Now, another scientist, Severo Ochao, he is a scientist. Again, he worked on the synthesis of the RNAs and also protein synthesis. He used an enzyme, and that enzyme is Ochao enzyme, that is polynucleotide phosphorylase. By using the polynucleotide phosphorylase, he could able to synthesize RNA. Was also helpful in polymerizing, synthesizing RNA with defined sequence. They know the sequence which is present in the RNA. Defined sequence in a template independent. Actually, if the RNA is coming from the DNA, we call it as a template dependent because the DNA is acting as a template. But here they have synthesized this using an enzyme, and that enzyme is polynucleotide phosphorylase. So this synthesis of polymerization of the RNA happens in a template independent manner. That is what? This is the enzymatic synthesis of RNA. Now, they could able to synthesize RNA, and the sequence of the RNA they know. Finally, by using the sequence of the RNA, what is the sequence of the amino acids that are coming in the structure of the protein? By comparing that, finally they could able to construct checkerboard of genetic code, which is here. The finally prepared checkerboard of the genetic code is here. So, this is all about, we can see 64 codon. Codons are the triplets. That means 3 nucleotides. See, 3 nucleotides. So, here, let's understand the salient features of each and every codon that is present in a genetic code. The salient features of the genetic codes are as follows. Codon is triplet, which have 3 nucleotides. Out of 64, 3 codes for nothing. They will code for no amino acids, and that is what we call them as a stop codons. 61 codons code for amino acids, whereas 3 codons do not code for any amino acids, hence they function as a stop codons. They function as a stop codons like UAA, UAG and UGA. These are the 3 codons which codes for no amino acids. They do not code any amino acid, and that codons will act as a stop codons, and we call them as a stop codon. Now, 1 out of 64, this 4 no, in that 3 will act as a stop codon, 1 will act as a start codon. This start codon, it is going to initiate the process as well as it is going to code for another amino acid, and that amino acid is methionine. Now, some amino acids are coded by more than 1 codon, and that type of codons are called as degenerative. Degenerate codon. What is this degenerate codon? See, for example, in the starting cell itself, we can see phenylalanine has been coded by more than 1 codon. See, U, it is a codon which codes for phenylalanine, and also UUC can also code for the same phenylalanine. That is what we said, that some amino acids, say for example phenylalanine in this case, are coded by more than 1 codon. UUU is coding for phenylalanine, and UUC is also coding for phenylalanine, and that property of codon, genetic code, we call it as a, so hence the genetic code is degenerate. It is able to code where some amino acids are coded by more than 1 codons. Now, the codon is read in mRNA in a contiguous fashion, that is continuous without any punctuation. For example, CG, CG, G, something like this, will code like 3, 3, 3. It will be read in a continuous fashion. There is no punctuations at all. After 1, we are giving comma, R, full stop, Nothing like that. Continuously it is going to read the nucleotides that are present on the mRNA and it is going to place the amino acids, that is the amino acid sequence will decide the structure of the protein. Now, the code is nearly universal with a very small exceptions where in the first example itself we said no, UUU code for phenylalanine. UUU will code for phenylalanine in the bacteria, even that UUU will code for the phenylalanine in the humans itself. It is universal. No matter what organism, it is going to be coded. Everywhere, seeing that codon will code for that particular amino acids itself. So, for example, from bacteria to humans, UUU would code for phenylalanine itself. The same thing, anything like CCA will code for proline only. CUC will code for leucine only. AUC will code for isoleucine only. GUC will code for valine. Similarly, GCC will code for alanine. GCC will code for alanine in bacteria. Same GCC will code for aniline in elephants also, dog also, human also. No matter the organization, the codon will be universal, nearly universal with a very small exceptions. Small exceptions to this rule has been found in mitochondrial codon, where in mitochondrial codon we'll see 4 stop codons and in some protozoans. Other than that, mostly that is what we mentioned, nearly it is universal. Nearly it is going to work universally. Now, as I already mentioned, out of 64, right, 3 are going to act as a stop codon and 1 is going to act as the initiator codon, and this is going to code for methionine 2. That means it is performing dual function. And that codon is AUG. AUG, it is acting as a start codon has dual function. It codes for methionine. It codes for methionine, and it also acts as a start codon, which is otherwise called as initiator codon. Initiator codon. This is all about 1. The 3 mentioned as a stop codon, which will not code for any amino acid. UAA, UAG, UGA. UAA, UAG, UGA. These 3 codons are stop codons, are terminator codons. Now, so this is the genetic code checkerboard, where this will give an information about, see, for example, UAC. If this is a codon which is having 3 nucleotides, we need to know which amino acid is going to be coded by this codon. And now, using this genetic code checker box, we have to find out that amino acid which has been coded by this codon. What is the codon we have? UAC in this example, right? This is the first letter, this is the second, and this is third. As we are having 3, we call them as a triplet, right? So, first U. Search for the first letter here. It is U, right? So, this box. Second letter here, right? And that is A. U, A. UA, 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 UA. Third is C. Third letter will be here. We have C, no? C. Here. What we have? U, A, C. U, A, C. Tyrosine. It codes for tyrosine. So, if we have codons, by using this checker box, we can able to find out the first letter, second letter, third letter, and finally it is going to give an information about which amino acid that codon is going to code for, right? This is how we'll check this genetic code. Now, so here, if following as a sequence of nucleotides, we have a sequence of nucleotides. This is in mRNA, right? In mRNA, predict the sequence of the amino acids that are coded by it. By using this, just now I have explained how the codons can be checked to produce, to know which amino acid it is going to produce, right? So, this is a sequence of mRNA we have. AUG, right? AUG. First letter A, second letter U, and third letter is G. So first letter A, U, second no, U. So this, and this is third letter is G. G. It is a start codon. It's going to code for methionine. So this is methionine. We have to read the codons on the mRNA continuously without any punctuation, right? Second one is U. First one, phenylalanine. Okay, UC. Phenylalanine. Phenylalanine, phenylalanine. Right? It is very easy by using the nucleotide sequence on the mRNA, by using this genetic code checkerbox, we can able to identify the sequence of the amino acids. Now, try the opposite. That means, consider that we have the sequence of the amino acids, but we have to find out the sequence of the nucleotides that are present on the mRNA. Just an opposite to this exercise. Following is the sequence. This is the sequence. Following is the sequence of the amino acids coded by an mRNA. Predict the nucleotide sequence in the RNA. This is the amino acid sequence, and we have to give the sequence of the nucleotides that is present in the mRNA. Okay, methionine. We can check here. Methionine. We have only one methionine. Yeah. A, U, G. Right. Next, phenylalanine. If that comes to phenylalanine, which code we have to write? Whether it is UUU or UUC. UUU or UUC. The same thing happens with other coming up series of the phenylalanines too. Here, we can come to the conclusion that this genetic code, as it is degenerate, that is more than one codon codes for a single amino acids. Some amino acids are coded by more than one codons. In that way, it is tough to determine the sequence of the nucleotide by using the sequence of the amino acids, but it is possible that we can know the sequence of the amino acids by knowing the sequence of the nucleotides that are present on the mRNA. That is a conclusion. So this way it is possible, but this way it is not possible because of the property of the genetic code. That means the genetic code is degenerative, right? Yes. Yeah. That is it for the today's session, where we have discussed about the genetic code, which is going to be helpful to understand the process of translation. In the coming up session, let's discuss further about the process of translation. Until then, keep learning with We Learning. Thank you.